No, green.
Okay, yeah.
Are there any pens?
Oh, this is a great question. There's lots of little tiny gold pencils. Wow.
Oh, thank you. What is your movie?
It's a automatically generated, like AI generated PowerPoint intro that I got from an old version of PowerPoint. But this feature doesn't exist on PowerPoint anymore, but it used to. Great question.
Not a real tree.
Nope. Well, I mean, I don't know how PowerPoint generated it, but there were like automated, like AI generated PowerPoint formats, like probably like 4 years ago or so. Yeah, so we used to be able to make stuff like this from PowerPoint.
I love training. Oh, I was like, wow, what city is this training?
And a lot of my like people do PhD and postdoc, but like a lot of my research before kind of had like pertaining to like infrastructure and stuff. And so I was like, oh, this is like vaguely related.
Cool.
Yeah.
Well, I guess I can go ahead and get started. We can go ahead and get started. Should we? Okay, so I'll start us off with a quick land acknowledgement. Thanks everyone for coming. So thank you so much for coming to Popping the Science Bubble. We're so excited about this partnership. I'm going to read our land acknowledgement and then I'll introduce the program. So Berkeley Public Library buildings are built on the territory of the Huchion within the homeland of Chochenyo-speaking Ohlone people. We acknowledge and honor the Ohlone people who have lived in Berkeley and throughout the East Bay since time immemorial. The library recognizes that the Berkeley residents have and continue to benefit from the use and occupation of unceded stolen land, and we pledge to recognize the permanence and the rich contributions that the Ohlone people continue to bring to the city and throughout the East Bay. We celebrate the strength of the Ohlone people and their culture. The Berkeley Public Library is committed to helping create a more just and anti-racist society and to furthering the visibility and celebration of Ohlone authors who are specific to the East Bay and Ohlone culture in our collections and programming. This is a living document that was co-written by Ohlone cultural leaders Vincent Medina and Louis Trevino and the Berkeley Public Library. And if you want to learn more about Ohlone culture, every third Wednesday of the month, we host a library program at Cafe Ohlone. It's part of the Hearst Anthropology Museum, and it's just like off to the side there. It's at 3 o'clock, and there's one happening tomorrow. You register online, and it's pretty cool. There's, you can taste some native food and learn a lot about native culture. All right, so welcome to Popping the Science Bubble. This is a monthly seminar series that aims to share new research findings from grad students and postdocs at UC Berkeley with the general public and create constructive discussion about a variety of science topics. We have 2 speakers at each seminar who will talk about their current research or a topic that they find really interesting. The organizers are 2 graduate students at UC Berkeley, Kaylin Zwang and Daniel Brett-Hauer. This program happens every 3rd Tuesday of the month at 5:30 p.m. at the library and on Zoom. If you're interested in checking out their past seminars, you can visit their website and sign up for the listserv to keep in the loop for upcoming seminars. Also, check out their YouTube page to view this lecture and see an archive of past lectures. All right, all of the Science Bubble team introduce tonight's speakers. Thank you.
Thank you, Kelsey. And hi, everyone. Welcome, and thank you all for being here. My name is Kaylin, and we're very excited to have Lauren and Amir today as our speakers. Like Kelsey said, feel free to, if you're interested in hearing more about science research happening at Cal and the Bay Area, many institutions in the Bay Area, feel free to sign up via our listserv that we have a QR code at the end, or if you can Google Popping the Science Bubble and it will show up. And so, and also feel free to visit our YouTube page and our website with a huge archive of all the past science talks dating until I think from 2020. So I'm going to introduce our first speaker, Lauren Walters, originally from Maple Valley, Washington, a suburb outside of Seattle, Lauren spent college and grad school in the Midwest battling cold winters. Back in California, Lauren's been enjoying working at a Berkeley lab on robots, chemistry, battery research, while hiking and walking all the sun that we have here. Her main hobbies include running, hiking, and spending time with friends, but she can also be persuaded to play some video games. And she also enjoys planning vacations and looking up flights that she doesn't actually book. So take it away.
Thank you for the great introduction, Keelan. So for those of you who might be listening, my name is Lauren Walters and I'm a researcher at LBL. And if anyone hasn't grabbed a crossword yet, you can take notes on my talk with that crossword. But I'm here today to talk to you about some of my research that's pretty near and dear to my heart. And this is really what happens when we want to make materials, and also what happens when materials degrade. It's something that we should really care about because it pertains to things like energy and batteries and infrastructure. And I think one of the best ways to understand materials degradation, materials chemistry, and what happens when new materials form, or at least one of the most interesting topics to do this would be to think about what happened in Flint, Michigan. And so if any of you were paying attention to the news in approximately 2016, you might remember the fact that it was reported that kind of almost out of nowhere, there were really, really high levels of lead in the water in Flint, Michigan. And so, you know, a lot of our drinking water does have small amounts of lead, but in Flint, Michigan, we were getting upwards of 13,000 parts per billion when we see that the cause for concern is 5 parts per billion. So that's like 10,000 times, or yeah, about 10,000 times more lead in the water than there should be. And that's really horrible, in part because, you know, lead is bad for everyone, but it's actually particularly bad for children. It can impact, you know, how children grow and how their brains develop. And so it's something we want to prevent. And this happens because there is infrastructure degradation. It's chemical reactions that allow lead to reach into our water system. But actually one of the worst parts I think about this news and about this being covered is actually the fact that it's not an isolated incident, right? We can see reports of this throughout the news, for instance, in Washington, D.C., also in the 2010s. What's really interesting here is actually, it kind of shows that this is a science problem. So if you look at this graph kind of farther on the right-hand side of the screen, the red line here, I'll use my arrow. The red line here shows levels of lead, and this right here is when the government was kind of aware of the fact that there was lead in the water. They tried to intervene, but as you see in the preceding years after they intervened, the amount of lead actually went up. And I think this goes to show that there isn't kind of a one-size-fits-all solution, and we need to be aware of the chemistry that happens to allow there to be these massive failures. And you might say, well, maybe we can intervene, but at the end of the day, there's actually like upwards of 10 million active lead service lines in the US that like pipes everywhere. And so, you know, one of the things we want to do is understand from like a chemical perspective, why is our infrastructure degrading? You know, what's happening and how can we prevent it, right? Because we all want to be able to drink clean drinking water. But just so that I don't scare you, this ends up being a problem that's actually a lot more prevalent back east. So I think our Berkeley water is probably a little safer. And so I did forward this news article, but if you didn't have a chance to read it yet, would absolutely recommend reading it because it kind of puts into context how, you know, widespread of an issue this can be. And so in order to understand lead and lead problems, but also, you know, material synthesis and chemistry, what we'll first do is I'm going to lead you on a 5-minute science or chemistry lecture. So please stick with me through that. We're going to go over phase diagrams and try and apply them to lead pipes. And then afterwards, we're going to use this as a roadmap for chemical synthesis and a lot of my research that actually takes place in LBL right now. And then finally, we're going to use kind of an outlook for synthesis science and how we might be able to make the world a little bit of a better place by understanding how materials form. And so the best way, you know, as a chemist, a material science, when I think of chemistry, making new materials, I really think about baking. I'm not actually a baker, but if you think about kind of the key steps of baking, so I'm sure maybe show of hands, has anybody baked anything? For sure, right? So there's kind of 4 key steps of chemistry, 4 key steps of baking, right? The first is that we're going to gather our ingredients. So we're going to get our sugar, our eggs, our flour, maybe some butter. And then after we weigh them out, after we gather them, we're gonna mix them and we're gonna put them in the oven, right? And we're gonna put them in the oven for a certain amount of time. And then after we get all of the baked goods we love, right? Like croissants and cookies and cakes. And honestly, chemistry and science is a lot like this. And so when we think about chemistry, instead of having ingredients, right, we have atoms. And these are basically the basic building blocks, not only of ourselves and, this world, but also of the field of chemistry. And so when we think about making new materials, right, or chemical reactions, we're really thinking about combining different atoms together. And so a lot of the names of atoms might be familiar to some people. So it's going to be things like lead, chlorine, argon, phosphorus. But at the end of the day, when we are kind of performing chemistry or performing chemical reactions, you can kind of think of it a little bit like baking, right? You're taking ingredients, which are atoms, you're combining them together into new arrangements, and you get a new thing out. And so there are a lot of different reactions that might be familiar to people, or non-reactions in some cases, right? So when we think about kind of key components within chemistry, we could think of what happens when you mix oil and water. Probably nothing's going to happen. Or you might mix salt with water and you might be able to dissolve something. But again, at the end of the day, I want you to think about chemical reactions when we make new materials as we have ingredients, or when we have chemistry, right? That's going to be an atom or a chemical combining together, potentially heat it up, and then we're going to make a new material. And so in the case of iron and oxygen, for instance, if we combine these together, we often get what we consider to be rust, right? So if you leave a nail, for instance, outside, you get rust at the end. And so some of my favorite chemical reactions actually are the classic gas creation from vinegar and baking soda. And this is actually the formation of carbon dioxide. You can also think about chemical reactions in terms of electron leaving a sodium atom. So anytime you put, for instance, the more chemical or the more science-oriented people here, anytime you put, for instance, a solid alkaline metal into water, it actually has a pretty explosive reaction that occurs. And then probably more pertinent to this talk is going to be the formation of rust. And so that's going to be whenever the outside of a metal oxidizes. So the metal reacts with oxygen to form a rust on the outside. But similar to baking, how do we know when a chemical reaction will occur? Hi, welcome. If you all want to take notes on my talk, you can grab a crossword puzzle. They're somewhere around here. So who all has seen the Great British Bake Off before? Any audience reaction? Is that everyone knows what the hardest challenge is, right? The technical challenge, where they basically say, make something, right? And usually it's something like, I don't know, make a shoe pastry, but then, they'll give basically the name of the baked good. Potentially there's some type of description. That's it. That's all they give you. And for a lot of the participants, right, in this baking competition, even with minimal instructions, it's really hard to recreate what the judges are looking for. It's really hard to create a good product. And similarly in chemistry, when we're trying to make like a new material, right? When we're trying to make and we don't have a recipe, we don't have a method, it's really challenging to make something. And so that kind of brings us to, when we think about this from a chemical perspective, how do we know when a chemical reaction will occur? How do we know what we're going to make, right? And there are huge fields of this, catalysis research and thermodynamics. But at the end of the day, as somebody who thinks about most things from a thermodynamics perspective, it's kind of a study of energy, I tend to think of chemical reactions happening due to energy. So all this means is that, when we think about dropping a ball, right, it's going to go down because or when we think about a ball rolling down a mountain, typically it's going to roll down and it's going to kind of decrease the energy of the system. And so energy, at least, is going to be decreased every time a chemical reaction occurs. And in a lot of cases in our world, whatever material forms is going to be basically the lowest energy material, right? And so when we're trying to think about, what's going to form, how can we create a chemical recipe? How can we understand all of these reactions, really we think about it in terms of energy. Where if we were a ball rolling down a mountain, what's going to reduce our energy the most? And, again, similar to if we're in the Berkeley Hills trying to get to the lowest energy part of our system, a lot of people, at least in the aqueous world, the aqueous thermodynamics material science world, will do things like create phase diagrams. And all this means is that we're mapping out the environment. So for instance, if you change temperature, if you change pressure, in this case, the environment that we're changing is voltage and pH. If you change these different parts of the environment, how does that change with the most energetically stable material? And so we can map out actually with this thing called the Courbet diagram made by a French scientist. We can actually map out when we change different parts of our environment, what's most likely to occur. So this is basically mapping out copper in this case, not lead yet. But if we have copper in water, if we have it at a low voltage, we're basically just going to recover copper. If we have it in certain pHs, we might dissolve the copper in water. But all this is basically it's a map. It's a roadmap to a recipe. It's a roadmap to a chemical reaction that tells us what material is most likely to form. And so as a recap again, I know that was a lot of chemistry, it was a lot of material science, but you can think of chemistry like baking. You put in the ingredients, usually you do something like mixing, or you can do something like increasing the temperature, and you get a different material back out. This can happen, for instance, with a metal, when we combine oxygen to get rust. And we often understand which materials we're most likely to get out through, for instance, making phase diagrams, making maps. So if we apply this back to this original problem, right, like why do we get so much lead in the water? Why all of a sudden Flint, Michigan has so much lead in their water after such a long time with having, kind of like stable pipes, for instance? First, you need to understand why the lead got into the water in the first place. And unfortunately, some of this kind of gets back almost to a social justice issue. Often people will think when there's lead pipelines, we need to replace them. But when that happens, what usually happens is that this lead gets basically disrupted, right? So it was sitting nice, it was a nice sediment at the bottom of the pipe. Then as soon as you start extracting one or two pipes at once, all of the lead that was sitting there comes up. And because there's like increased surface area, it basically solubilizes and moves back into the water system. And so that's really, really bad. And so often by trying to replace pipes, we're actually increasing the amount of lead in the water, which can be really, really frustrating. But one of the great things is that you can actually try to decrease the amount of the lead in the water by trying to have rust on the inside of our pipes, right here, some type of oxide barrier. So again, if we look back at our Courbet diagram, one interesting thing we see right here is that this is a solid, right? Solid. So there are parts of our map that are kind of solid rust on the inside in our pipe. And the idea behind that would be that if lead is solid, it's not leaching into water, it's not soluble. But if we have tap water, just at kind of regular conditions, we're going to be right here. That means that the lead is actually soluble. So that means that the most favorable chemical reaction, the reaction that's going to lower the energy the most, is actually solubilizing lead into water at ambient conditions, so to speak, in chemistry speak. But what we can do in order to increase how much rust, how much oxide we're going to have in our lead is actually adding additives. And in this case, the problem with Flint, Michigan wasn't that they were replacing their lead, but that they stopped adding additives to their lead. And immediately, what would be the most energetically favorable material are lead ions leached out into water. And so I'm going to move, so long story short, keep adding additives into water to make sure if you have lead pipes that you keep having rust in your pipes instead of soluble lead. One thing that I want to do though is kind of transition a little bit. So we're thinking about materials, we're thinking about what materials are most energetically favorable. And we can make other types of thermodynamic maps. I just want to point that out because, synthesis science is hard. We're trying to synthesize new materials. That means to make new materials of chemistry instead of looking at degradation. And this is really important because, I'm a battery materials lab and I work at LBL. And that is a Department of Energy lab. And so we really care about making new materials for things like batteries. But what we found is AI has been predicting a bunch of new materials we should be able to make, but we haven't been able to make them yet. Because if you remember Great British Bake Off, right, it's really hard to make recipes for new materials we've never made. And so what we did is we actually decided to create the robot lab And so what this does is it just automates sample preparation for us. My video is being a little slow. We'll see if it plays. Oh, it's so much cooler when the video plays. All right, well, we basically have a number of different robot arms that will basically go and it will combine different powders together. Again, think right back to baking. We're going to combine different powders together. We're going to move them into our ovens that you'll see here and heat them. And then we have kind of recovery stations at the very end. And oh, some of the videos are playing. But the whole point here is basically, you know, synthesis is really hard, chemistry is really hard. It's totally trial by error in some cases. But there are other things that we can do to hopefully speed up this process, such as making a lot of these thermodynamic maps, these energy maps, to really understand ahead of time how we make recipes and how we understand what materials are best to form. And so, oh, those videos were hard on my computer. Sorry, y'all. So anyway, just kind of wrapping up kind of some of our contents, right? Chemistry is really cool, right? Similar to baking, you know, you can combine also a lot of the same things together and get a different material back out, right? Maybe you're going to get bread, or maybe you're going to get a croissant, or you'll get a cookie. But at the end of the day, you know, there are really, really important problems that we can think about and solve, including the fact that we can understand from a chemical perspective and an energetic perspective, maybe why our infrastructure might be failing, and come up with real solutions for how to make sure that we have, you know, a little bit of a healthier and safer world. And one of the ways, at least at LBL, that we've been trying to do this is by trying to automate some of these tasks that would be really hard, again, like recipe development for new chemicals, so that we can get, you know, again, kind of a cleaner and healthier world. In this case, you know, my lab is really cares about batteries, but from a material science perspective, we also care about things like solar panels and pharmaceutical and, again, infrastructure as well, having clean water. And so with that, thank you so much for your time and attention, and would love to take any questions.
I'm curious about these additives for lead. I've never heard of them before.
Oh, yeah.
Whose responsibility is it to add them to the water system?
Yeah, great question. So even maybe a little bit more of a background on Flint. So I think that Flint is similar probably to a lot of suburban counties where they were on Detroit's water system. And so they were part of this massive utility conglomerate that would, it kind of probably similar to PG&E or any other utility conglomerate. But then to save money, they ended up outsourcing to a basically small private water company. And then that, and theoretically, any company that's providing water would, I guess, probably work with the government or something. But as far as I know, there's not like a whole lot of regulation on it. And so they ended up, because they had lead pipes, but they didn't have the phosphorus additive from their original water supplier, all of a sudden, again, the lead concentration exploded because no one was adding it. But what happened was that it's up, I believe, to the utility companies. There is, I think, a little bit of government regulation, but not much. And it's kind of up to like whatever your utility jurisdiction. Yeah.
So the additive is a phosphorus?
So it depends. So I think that's actually also up to your county. Typically a lot of people will use phosphorus. What ended up working for Flint was phosphorus, but it's actually, so one of the reasons I brought up DC is that they actually had a different issue, and that's part of the reason why their concentration went up, was because they tried adding phosphorus, but the problem was that their lead was actually coming because they added chlorine, similar to swimming pools, to decrease the amount of bacteria in their water. But then the chlorine increased, the water has like a dielectric constant, the voltage, and that increased the voltage so much that the phosphorus wasn't effective anymore. Water chemistry is really, really hard. And so it just kind of goes to show, right, there's so many different competing reactions that, yeah, phosphorus is really, really common, but you have to be aware of what else is in your water. And again, since it's not, I don't think there's like a standard, it's not regulated. Yeah, a lot of things going on.
Can you tell us a little more what this robot is doing? So what like materials are you trying to synthesize specifically? How does it relate to like the batteries and
Yeah.
this stuff?
This video looks like it's playing a little better. No, it's not. Oh, shoot. So my lab is battery lab.
Mhm.
We're really interested in making solid state batteries. So that means that instead of having you have an anode, an electrolyte and a cathode, and that middle part of your battery has like historically been a liquid. It allows ions to move back and forth when you charge to carry electricity or to hold electricity. Our lab is really interested in solid state batteries. So we typically are making cathodes, but also a lot of solid state electrolytes because, you know, ions are pretty large often and trying to move them through a solid is much harder than moving them through a liquid. And so in this case, this robot, as the video goes and then pauses again in different steps, is basically doing a solid state synthesis with chemistry. So it's typically combining things like phosphates, oxides, silicates, so different materials together that would be able to make the different components of a battery. And so first what it'll do is weigh out powder. It actually adds ethanol to it, to create this kind of like liquid slurry mixture, dries it, and then it heats it. And then what it'll do afterwards, and so the robot will carry the sample through each of the steps. And then afterwards, it will actually also extract the powder out of like the tray, the crucible we heated in. And then it'll actually do different tests on the material to try and figure out, for instance, like what the material is, if it has good properties, things like this.
So at the beginning, you also mentioned lead can also slip into water when you replace the old pipes. What are some strategies to prevent against that? Like, because you know, additive probably wouldn't.
Yeah, again, I think people will put a bunch of phosphorus in the water, but that also has like a lot of challenges as well. I think what the typical recommendation can be is just to replace an entire service line at once. But then I don't know. I think that that's a problem if there isn't a good strategy because you can't just turn off someone's water for a month. Right. And it's hard because when you do it like as a one by one process, like it disrupts, it pulls that rust kind of scale. We call it a scale in the like water science community, but it pulls that scale off that's protecting the lead from coming out. Yeah, I'd say replacing everything at once. Yeah, I'm not totally sure if I'm being honest. Well, thank you so much for your attention, and I think it might be Amir's turn then.
Let's give another round of applause.
Oh, if anyone wants the answers, I have them.
While Amir is getting up, I'm going to go ahead and introduce him. So Amir is a Jane Coffin Child postdoctoral fellow at Berkeley, co-advised by Susan Marcasi and Carlos Bustamante. He's originally from the Boston area, where he did a PhD at Harvard with Eugene Shoknovich, working to develop simulation approaches to predict how proteins fold up in their specific 3D shapes that let them function, the topic of this very talk. He is now developing experimental approaches to test these same questions. Outside of lab, Amir is an avid musician and especially a composer. Amir has written a number of science-inspired compositions that have been premiered in various countries around the world, including Paris, Bogota, Colombia, and Boston. He also loves hiking. The Bay Area is such a beautiful place for it, and running and listening to podcasts and learning all about diverse topics. Sounds like we'll be learning ourselves a lot about diverse topics here as well, brought with a wide range of everything that you can do. So please take it away.
Hi, thank you so much for the introduction. I do just want to make sure that the audio is gonna work because it's like not responding again. What do we do again? Oh, it's it. Yeah. Lauren, in addition to being a great material scientist, is also an expert at dealing with technology. That's we've learned. Oh, it seems to be fine, so maybe it's fine. I just okay, guys, sorry. Thanks. Can the Zoom see my screen?
Yes.
Awesome.
It is your
Yeah.
presenter.
Oh, but it's in presenter mode, so we can see like.
No, I don't touch that. Then we're gonna have to swap that. No, if I press swap display, that's actually not.
There we go. That now.
No, this is not. This.
Oh.
I'm gonna stop and share.
Under more, can we maybe highlight?
I think what I have to do, I have to share my desktop instead of
Yes.
sharing the PowerPoint.
Yes, please do that.
Instead, now it's more. What about now? Okay, now they can actually see the full. Yes, that's awesome. Hi, everybody. It's great to be here. It's really an honor to have been invited to give this presentation. I'm very happy to see a couple of friends from many epochs of my life here, as well as a few friends and family on Zoom. So thank you all for coming. And I'm very excited to tell you guys a little bit about my research. And I have titled this talk, "The Dance of Proteins in the Cell." I'll tell you all about what this means, why I chose this title, and you're already starting to see some allusions to music. So as was mentioned, I am a composer, and as the talk goes on, we're actually going to see that these two things will come together, the proteins and the music. So stay tuned. So my talk is going to be all about proteins. So proteins are, of course, something that we eat every time we have one of our meals, but there's a reason why we consume proteins, and that's because they are actually what do basically everything that we associate with life. So let's think about, what living things do. Living things move, right? So, we have a runner here. I'm actually training for a half marathon myself. It's been a little brutal, but, we're going a little at a time. And motion is made possible by protein molecules, right? Every time your muscles contract or expand, that actual contraction is made possible by proteins such as myosin. Here we've seen sort of a few, images of experimentally solved structures of protein molecules that mediate the ability of organisms to move and muscles move, right? As you're sitting here, hopefully understanding my talk, you are processing information, right? And, cognition is very much linked to decision making as well. And this is also made possible by proteins, right? Because our neurons have to conduct electricity, and it's protein molecules that actually allow, ions to come into your neurons and travel as electrical waves, and then proteins actually sort of allow neurons to sort of do calculations depending on the voltage that they receive. And so, one other thing that all living things do is they metabolize nutrients, right? They break down macromolecules into smaller molecules that they can use, and then they use that for energy production, and proteins play a crucial role in doing that. You have enzymes that actually break down macromolecules in food, and then you have some specialized proteins like this one that actually, they kind of spin around like a whirlwind. They're really cool. They actually use, the flow of certain ions in order to produce molecules that your cells can then readily use for energy. Finally, a key aspect of all life is growth and reproduction, right? And things like copying all your DNA, allowing your cells to go from one cell to two cells, all of that is also done by proteins, right? So we see that proteins are just everywhere. They do all kinds of things. They're just indispensable for life. So how do they possibly work? And sort of the short answer for how proteins work is they work just like any machine or anything we're familiar with from our day-to-day life, which is they need a structure, right? Across the bay here, we have the beautiful Golden Gate Bridge. I actually took a bike ride across the Golden Gate a couple days ago this weekend, and I found it so cool to sort of notice the suspension cable. Actually, it's a little foggy, so I couldn't see the top of them, but seeing the big posts sort of as I was approaching the middle, it was so fascinating and kind of see it from the inside, and it really made me appreciate how its function is dependent on its structure, right? It was built just right to allow it to handle, a specific load and, allow cars and other vehicles to actually go across, right? So protein function, just like the function of day-to-day things, actually depends on the three-dimensional, now molecular structure of proteins as opposed to macroscopic structure. And it turns out that just like, if you don't build a bridge correctly, it can collapse. When proteins have not attained their correct structure, they can also cause different types of catastrophic failure for the cell. So there are a number of what are called protein misfolding diseases. Here are only a small subset of them, but some of the arguably deadliest ones. So cancer, for example, about half of cancers are actually estimated to be caused by different proteins that lose the ability to kind of regulate our cells' ability to divide. And as a result, your cells divide sort of uncontrollably. And many times that's actually because the proteins that are sort of the gatekeepers to cell division lose the ability to do that, and they don't get the right structure, and this causes the issues associated with cancer, right? Alzheimer's disease is caused by accumulation of incorrectly structured proteins in the brain that are very damaging to the brain. Likewise, Parkinson's, cystic fibrosis, type 2 diabetes. There are many, many that I haven't even mentioned here. So, it should then become clear that a very important kind of question for life is what ensures that in healthy, normal conditions, proteins acquire their correct three-dimensional structure. And why is it then that in disease, this process goes wrong? So that hopefully this knowledge can allow us to rationally design therapeutics to address these ailments and ensure proteins actually get their correct structure. So I'll take us on a little bit of a tour into molecular biology so we can kind of start to answer this question. When we think about macroscopic structure, we always think about a blueprint, right? The blueprint is what actually tells the people in charge of the construction how to set everything up in order to produce a functional structure. In the case of proteins, the blueprint is our genome, right? Our DNA. The ATCs and Gs of our DNA are actually what give our cells the instructions for how to assemble a protein. So a protein is made up of amino acids that are kind of strung together. I've depicted them here. You can think of them as kind of beads on a string. There are 20 sort of types of amino acids. Each of those 20 amino acids has kind of its own chemical and physical properties. And depending on what DNA letters are present in a sort of given location in a gene, which is like a message or a blueprint to build a given protein, that will dictate in what sequence these amino acids need to be strung together, right? And then, as I mentioned, different amino acids have different, properties. Some are positively charged, some are negatively charged, some are kind of greasy or hydrophobic, so they don't like to expose themselves to the water that actually surrounds all proteins in our cell. And somehow, as a result of all of these interactions, we'll talk about that a little more in a bit, we end up with a protein that's actually folded, that has actually kind of wrapped itself up around in space in some specific way, such that that molecule can go on and do something, right? So, for example, for the one I've shown here, you could imagine maybe some other molecule can fit inside this cavity and this sort of ring-shaped structure, and then by fitting in there, some chemical reaction might be able to happen, for example, right? And just to give sort of a sense of scale, proteins are actually quite large as far as molecules go. Each of these amino acids might have something like, 10 to 100 atoms within it. So when you string a bunch of them together, you end up with molecules that are 10,000 to about 100,000 atoms inside. So they're kind of large molecules, but that doesn't mean they're, large as far as molecules go, but that doesn't mean they're large. They are impossible to see even by light microscopy generally. So that just was to show all the sort of length scales that exist, many of which are so much smaller than what we can visualize. So in fact, we actually have different types of experimental techniques that let us figure out what a given protein molecule looks like once it's kind of correctly folded. It lets you resolve sometimes at the level of individual atoms where each one is in space. And you see here, I've shown some examples of protein structure. You can see that proteins actually range in size. Some, like this one on the top left, are really tiny, just a couple tens of amino acids in length. Others are gigantic, like this membrane protein here. It's this huge protein that actually inserts itself into cell membranes and sort of everything in between. And you can just appreciate they're really beautiful molecules. They're sort of very intricate. They wrap around themselves in all sorts of ways. And we actually have so many structures of proteins that have been solved by structural biologists over the last couple decades that we've been able to train machine learning algorithms, artificial intelligence algorithms, to take in the genetic sequence of ATCs and Gs that encodes the protein and spit out what that protein actually folds into, kind of when all said and done, assuming it's folded correctly. So the algorithm that sort of is most famous for achieving this task with remarkably high accuracy is the AlphaFold algorithm. Initially AlphaFold 2 that was released a couple years ago. Now there is a new version called AlphaFold 3. So this is an algorithm that sort of achieved something like greater than 90% accuracy in sort of being able to predict the three-dimensional fold of a protein just given its sequence. And so when this was released, there were all these headlines by DeepMind, which is the company that made the algorithm, and by other companies saying that the protein folding problem has been solved, right? AlphaFold solves protein folding, AlphaFold solves protein folding using AI. I would argue, maybe I'm biased, right? Because I need something to work on, right? But I would argue as a protein folding scientist that these headlines are all quite exaggerated. In the sense that the protein folding problem, which is sort of saying, how do we get this three-dimensional structure from sequence, has many parts to it, right? One of which is the part of actually predicting the structure, right? And AlphaFold does an amazing job at that, right? However, there is another question that AlphaFold actually doesn't address at all, and no artificial intelligence algorithm addresses this at the moment, which is how this happens, right? How is it that starting from a chain, a sort of linear chain of amino acids in some particular sequence, does this protein actually find its correct structure as opposed to any other structure that a given amino acid chain could possibly contort itself into, right? And this is a major unsolved question. As I mentioned, it has implications in disease because when the process sort of goes wrong, it can cause various diseases. This is a question that I'm devoting my research to. So let's go ahead and, make some progress in understanding how this might happen. And I'll sort of give you a summary of this field, this question of how proteins fold. It's a field that's been around since about the 1950s. It was largely pioneered by American biochemist Christian Anfinsen, who did some seminal experiments in trying to address this question. And he had the hypothesis that proteins can fold entirely by themselves. So if you take a protein molecule and kind of stretch it out, and then let go of it so that it can go on and sort of wiggle, jiggle, do its thing, he hypothesized that just by virtue of the fact that different amino acids have physical properties, some of them are positively charged, some of them are negatively charged, some are greasy, hydrophobic, like to bury themselves away from water. From the sum of all those chemical and physical interactions, the protein would actually be able to fold itself, a little bit like this animation we see here, right? That is just an animation. It's not actually based, it's not a simulation based on any sort of physics. It's just sort of something I made kind of for fun. And it turns out that Anfinsen's experiments showed that this does actually happen for, he was studying one particular protein, and since then it's been kind of replicated for many other proteins. There are many proteins that just by themselves, just because of these interactions between different amino acids that they sort of talk to each other, allow the protein to sort of correctly and reliably fold. And this is a really cool process that's kind of akin to saying, let's take like a long string of yarn, right? The different sort of segments of the yarn, let's imagine they have like different chemical and physical properties, and sort of expecting that string of yarn to knit itself spontaneously into a sweater. I do have friends in the audience who actually knit, so I hope that my analogies are not too bad, but that's sort of the way I think of it. It's a really cool process that sort of this long string actually kind of folds itself up or knits itself. But as it turns out, this is not true of all proteins. So there's been some recent work by some of my colleagues at Johns Hopkins University that tried to address the question, what fraction of protein, if you sort of take the amino acid sequence, stretch it out and let it sort of do its thing, fold by itself, what fraction will actually fold up reproducibly and reliably? And they actually found that only about half of proteins of the ones they studied. So they studied proteins in E. coli. It's a type of bacterium. And you might wonder how useful is this given it's not humans? Does it tell us anything about human diseases, et cetera? It turns out that even though E. coli are quite far from us in the evolutionary tree of life, many of their proteins resemble, both in structure and in function, the ones that we possess in our own cells. So despite it being so, and it's an organism that's sort of very easy to work with, cultivate in the lab, yet it's quite useful for sort of studying properties of proteins, despite it being so far away. That kind of shows you how conserved a lot of life is, right? So among proteins in E. coli, which again are potentially not that different from the ones we possess, only about half of them, 54%, were found in this experiment to actually be able to fold up into the structure that they started off with before sort of they were kind of unwinded and given the chance to try again, right? A solid 27% of them were not able to do this. So if you actually took the protein, sort of unfolded it and tried to sort of let it do its thing, fold up again, it ended up more like what you would expect would happen if you have like a bunch of yarn, right? It's not actually going to sort of knit itself into a sweater. It'll more likely than not actually get tangled up, right? And then about 20%, the experiment was not able to conclusively determine one way or the other whether they can refold like this or not. So there's definitely at least a substantial fraction of the proteome, if not even larger than this red fraction of the pie chart, that cannot fold by itself, right? And obviously, this is going to potentially pose a problem for the cell, right? As I mentioned, it's crucial that proteins have that sort of very specific three-dimensional configuration to be able to function. So how do we actually go about resolving this issue? It turns out, so, again, I'm going to use potentially botched knitting analogy. So if you're a knitter, you're not going to unspool all of your string at once and start to actually knit with it, right? You have your ball of yarn, you're going to sort of take a little bit at a time, work with that, make some part of, I don't know, the sleeve or whatever you're making, then take some more and then sort of build up on top of that, right? It turns out in our cells, there exists a mechanism that might allow proteins to sort of knit themselves in an analogous way, a little bit at a time. And that is actually the ribosome. So people might have heard of the ribosome in sort of, whenever you took like a science class, but the ribosome is this machine, it's this huge molecular machine made up of both proteins and of RNA. That is found in all of our cells. There are gazillions of them in each of our cells. And the function of the ribosome is actually to read the genetic information, actually not in DNA, but in the form of mRNA. mRNA, we've heard about it a couple years ago when we, got vaccinated with the vaccines. It's sort of an intermediary between DNA and proteins, but it sort of has the same information practically as DNA, right? So the ribosome kind of scans the A, U, C's, and G's of the RNA and uses it to sort of figure out in what order should the amino acids be added to the protein. And it actually adds amino acids one at a time to the protein. So the protein kind of grows. You can see in this animation that's kind of been replaying that the protein actually kind of sequentially emerges from the ribosome. And this is actually where this analogy comes in, right? It turns out that for some proteins, as you can see just happened in the movie, they can actually start to fold one little bit at a time. As amino acids are added, there are sort of places where you've actually added enough amino acids such that, for example, a positively charged one and a negatively charged one might actually both be out of the ribosome and able to make contact, or other properties might be satisfied. This allows proteins to actually fold not all at once, the whole string of amino acids, but a few amino acids at a time. And this has been shown to actually significantly increase the odds, the likelihood that certain proteins fold correctly, as opposed to if you have the whole amino acid chain present all at once, all the amino acids at once, and sort of let it fold, right? We saw that some proteins can fold that way, but not all of them. And one thing you'll appreciate from this animation, again, this is not an animation that's based on any data. It's actually just, I made it kind of, as with an artist's interpretation, you could say, is you can see that here are tRNA molecules that actually add the amino acids to the protein. You can see the ribosome kind of scanning along the AU, C's, and G's. This is the mRNA here. And you see that it kind of slows down in some places. It doesn't kind of advance uniformly. It doesn't read the information uniformly. It turns out that this is actually something that's evolved for some proteins, where some proteins have evolved such that as the ribosome is sort of building it up, the ribosome won't go at a steady pace. It will actually kind of go in bursts and pauses. And this is to actually potentially allow certain portions of the protein enough time to be able to start folding, like we just saw there. Timing is so perfect. So, you can already start to see that there is sort of a music or a dance-like quality here, right? There is a rhythm, right? The rhythm of how quickly the ribosome kind of moves along and reads the information of the mRNA. And then there's the dance of the polypeptide chain, how it sort of starts to flop around in space. And so you'll see later that I've actually taken this analogy maybe one step too far and actually written a piece called The Dance of the Nascent Chain, the nascent chain actually being what we call a protein as it's kind of being built up, as it's emerging from the ribosome. But before I show you my music to sort of wrap things up, I'll tell you a little bit about the specific research that I am doing, which is actually trying to develop a detailed molecular picture of this process as it happens. Because again, this cartoon I'm showing here is just an animation, which is not taken literally by any means. I want to actually collect data that will allow us to actually visualize what's going on, which is then a first step. If we want to sort of design AI algorithms that can predict this process, we need to have enough data first, training data, right? And we don't have any right now because it's such a hard process to visualize. So one of my goals is actually to be able to see this kind of crucial process that the protein is being born, how it starts to find its shape. So I'll tell you about two techniques kind of briefly, and if there are questions later, I'm happy to elaborate on either of these methods, how I am doing this. So As I mentioned, as was mentioned in the introduction, I am co-advised by two professors at Berkeley. One of them is Dr. Susan Marketsy. The Professor Marketsy has sort of devoted her life to developing techniques that, a technique that's called the hydrogen deuterium exchange that can actually let you sort of take snapshots of a protein as it's sort of being born and as it's starting to fold into a specific structure. And I'm taking that technique now and applying it specifically to proteins on the ribosome, because in the past, it's a technique that's been used in proteins in isolation after all the amino acids are there, watching it fold. Now I'm actually, looking at this. So let's imagine that we take two snapshots from the movie I showed you before, right? And our goal is for each of these snapshots to sort of see what the protein actually looks like, right? Let's imagine that in one of the snapshots, the protein looks like this. That's the ground truth. We, of course, don't know that, but let's imagine that's actually what's happening. How are we going to learn that this is what it looks like? What we're going to do is we're actually going to sort of chemically label the protein. So we're going to take the protein and we're going to put it in a special type of water called deuterium oxide, where instead of hydrogen oxide or H2O that we're used to, and we heard a lot about very nicely in Lauren's talk before, this is a modified type of water where the hydrogens are heavier than normal. Hydrogen usually just has a proton and an electron. But deuterium has a proton and a neutron as well as an electron. So it's heavier than normal water. And deuterium will do this cool thing where if you put a protein in a solution with this D2O, this heavy water, the deuterium will actually chemically attach itself to certain parts of the protein sequence, to certain amino acids along the length of the protein. But it will only do that at amino acid locations that are not part of the folded structure, right? Only at amino acids that are actually kind of facing the solid, facing the solution surrounding them. And so if we were able to label the protein and then figure out what parts got labeled and which ones didn't, we'd be able to learn what was kind of exposed and what was sort of folded and be able to sort of build up a picture of what this particular snapshot looks like. And the way we actually do that is we do this labeling process for a short time, only for about 10 seconds, it turns out. Then we chop up the protein into little pieces. There are actually certain enzymes from the stomach that we use for chemical reasons. I can tell you about why we use those enzymes to chop up the protein into little pieces. And then we can actually kind of weigh each of those pieces using a technique called mass spectrometry, which lets you actually figure out for each of these little parts of the protein, how much deuterium did it take up, right? For instance, this part here, because it's kind of well folded, will end up not having taken up as much deuterium, and the mass spectrometry step will tell you that, whereas these parts here that are more unfolded will sort of take up more. And then from there, you can actually then figure out what the protein looks like, right? Which parts were folded, which ones were not. And we can repeat this at different times along the process of co-translational folding, which is what I've been describing, the process of how proteins fold as they are being translated from the mRNA code to the amino acid code, right? So this is sort of the main technique that I'm currently working on. But one of the other projects that I've sort of started to be involved in is using a totally different technique. And this is under the lab of my other advisor. So this is Professor Carlos Bustamante. He's a Peruvian biophysicist, also working actually one floor above Susan's in Berkeley, above Professor Marquez's lab. So, it's very easy for me to go back and forth. And what Professor Bustamante actually has developed is a technique where you use a very cool physical property of lasers and of certain materials, certain plastic materials. We're going to take a plastic bead and shot a very small plastic bead, micrometer size. So that's 10 to the minus 6, much smaller than you can see under the naked eye, but you can see it with a light microscope. And if you were to put it in a high-powered laser beam, it turns out that the bead has this cool property that it wants to stay at the center of the beam, at the focus point of the beam. And you can then take advantage of this to actually trap individual protein molecules, be they in isolation or as they're being born on the ribosome. You do this with a special type of chemistry that allows you to sort of connect the bead via what we call a handle to the protein itself on both ends. And it turns out that as this protein kind of folds and unfolds, or as it grows on the ribosome, it is going to exert tension on this whole system that's kind of going to tug on the bead. So you can imagine in a game of tug of war, if the sort of rope were to suddenly get longer, if you were to undo a knot as you're pulling, you would sense that, right? Because the tension would kind of temporarily go down. Or sort of if it were, if you imagine the rope started to sort of fold itself, form a knot, you would sense that it's actually more tension, right? Because the amount of slack you have gets shorter. So you could actually, by detecting what happens to these beads in the laser, you could actually sense tension over time and then be able to actually monitor how the protein kind of grows, how it folds, and so on. So that's sort of a brief overview of the type of experiments I'm doing. I know I went over that a little bit briefly, but I'm happy to sort of elaborate on any of that in the Q&A later on. And I just sort of want to zoom out before I conclude and kind of remind us, why we care about all this. Why are we sort of doing all of this, right? So as I mentioned at the beginning, protein folding, this problem I've been talking about all along, is the moment in which genetic information, this huge amount of information we have is the A, T, Cs, and Gs of our genome, becomes something useful, right? Because when a protein folds, it goes from a linear sequence of information into something with a 3D architecture that can go on and do stuff, can go on and do chemistry, can go on and do all these things that we associate with life, right? In my opinion, this in and of itself is a cool enough reason to care about protein folding, right? It's just fundamental to life. But in addition, it does have many implications, like I talked about at the beginning, in disease. It has other implications as well, right? It turns out that if we're thinking about how organisms evolve, including how viruses evolve, right? Like if you're considering different strains of SARS-CoV-2, the coronavirus, or if you're keeping your eyes now on the H5N1, on the influenza strains that might be emerging. One important thing about viral evolution is that for a virus to be able to actually evolve and take over the population, its proteins need to be able to fold up into something that lets the virus actually infect ourselves, right? And so being able to sort of predict what mutations will ensure the proteins that make up the virus can still fold up versus which ones will not is crucial towards being able to say which strains might actually emerge during evolution. Finally, there is a lot of interest now in designing new proteins that nature has never seen or never made before. AI has actually played a key role in this and actually being able to design proteins with novel properties. So this is a particular protein that was designed to actually be able to degrade plastic, right? We know that accumulation of plastic is a huge issue for our ecosystems, for our oceans. So if we had ways to better degrade it, that would be tremendously useful. So people were actually able to design a protein that if you mix it with a plastic water bottle and heat the system a little bit, it would actually able to degrade the water bottle into plastic monomer, which is crazy, right? And sort of being able to do this more efficiently will rely on not just sort of being able to predict folding, but actually be able to predict sort of how a sequence will fold, what fraction of molecules will fold versus misfold, having this kind of detailed knowledge, not just sort of this broad knowledge. So, just to sort of conclude, I'd like to thank you all for attending, and I will play just like a 1-minute clip of a musical composition that I wrote for piano I wrote it kind of during the pandemic, and it was first performed shortly after. This is performed at a very cool event that happened in Boston in 2022. It was an event at the Museum of Science called Music of the Genome, where it was a bunch of scientifically inspired new music compositions by myself, by other composers, some in Boston area, some from France and other places. And so I wrote a piece for piano called The Dance of the Nascent Chain. So the nascent chain refers, as I mentioned, to the protein as it's being born and starts to fold. And it's sort of, I use the boogie woogie dance as the type, the swing boogie woogie sort of music as the inspiration. And I hope you can see a little bit, these melodic melodies that kind of change in shape, these kind of traffic jams as you're right, with some kind of pauses and keep going. So I'll just play a little 1-minute clip of it, and then I'll take any questions. With that, I'll pause it, and if anybody likes new classical music and wants to hear more, many of my pieces are on my website, amirbitran.com, just my first name, last name.com. That I'd like to thank the organizers of Popping the Science Bubble for inviting me. It was so fun to be here. Thank you all for attending, and I would be happy to take any questions at this point.
So for the proteins that you or the research that you showed, some proteins can fold by themselves, but then there are a lot of them that cannot. So if you are there like categories of proteins that or features of the proteins that you know that like if they're by size or their functions that they're more likely to be able to fold by themselves?
Excellent question. So for people on Zoom, I'm not sure if they heard, the question was kind of what are the features of a protein that determine whether it will be in this kind of black category in the pie chart that it folds by itself versus in the red category that it actually needs some assistance to be able to fold. It's a great question. The authors of that study did do some analysis on these different properties. Things like charge actually affected this propensity, right? As I have to sort of remind myself what direction and when in, but I kind of my intuition would tell me, if I'm remembering it correctly, that a protein that is highly charged, has actually more difficulty to fold by itself versus one that's more neutral might be able to do it better. Larger proteins, as you were alluding to, generally have a harder time with this. Proteins with what are called multiple domains. So there are some proteins where it's made up of sort of subunits that are strung together. Each one kind of does its own function. Those have a harder time folding because the different domains might kind of get tangled up one with the other. And there are a few others as well. So there definitely has been kind of correlative studies linking proteins into one category or the other. And for most of them, there was a strong kind of correlation between the two. But we, still don't have a kind of deep predictive power with a new sequence will it kind of fold correctly or not. Yeah, great question. Yeah.
Yeah, thanks for the talk, Amir. I'm really interested in this question of kind of your analogy of the dance. Who's the conductor? How does the ribosome know to slow down and speed up?
Great question. So who is the conductor in this dance for how quickly the tempo of protein synthesis should proceed? It's something that I was debating if I should go into or not. It takes maybe a few minutes to explain clearly, but I'll just give you a quick answer and happy to elaborate. But it turns out that there is a hidden link. So actually, if you want to learn more about this, the organizer sent out a link to an article I wrote, I think in the email published in today's talk, where I talk a bit more about that question. It's this link called the Hidden Genetic Code, if you go to the email where this event was advertised. The idea is there's actually an additional layer of information in our genes. Besides encoding what sequence of amino acids the protein should be strung into, there is actually a layer that is believed to encode this. And it manifests in the fact that if you think about how amino acids are encoded, a given amino acid, say the amino acid cysteine, which is one of the 20, is encoded by particular triplets of DNA, and there are, if you actually count how many triplets there are, a triplet has, 3 positions, right? At each one, there can be 4 letters, A, T, C, or G. So it's 4 times 4 times 4, or 64 possible triplets. However, there are only 20 amino acids, right? So it turns out there is some redundancy in the genetic code. And for a while, it was thought that, so certain amino acids are encoded by multiple triplets. And for a while, it was thought that these are synonymous, right? That 2 triplets that encode the same amino acid, doesn't matter, you could use either one when you're encoding a protein. And it turns out, as we understand more and more, that that's actually not true. That different triplets that are supposedly synonymous have different properties, including different decoding rates on the ribosome. And there have been studies that actually show that there is conservation of sequences of slowly decoded triplets in certain protein sequences that are correlated with predicted folding intermediates, which I can tell you more about. It's a really fascinating correlative study that was done, and that's basically how it's done. Yeah.
I one, chaperones, question mark.
Yeah.
2, does the location of the folding matter? Like if it's folding into the ER, does that matter? Versus
2 excellent questions. So Sarah asked about a type of molecule that I was sort of debating whether to include or not, which ended up leaving out just because I didn't have time, which is chaperones. So chaperones are a class of proteins whose function is actually to help other proteins fold. I am very interested in chaperones because it turns out that they can actually collaborate with what I've shown. Where's my mouse? I can no longer see. So I can no longer seem to be able to go back on my slides for, it's because I think there's some slideshow and presenter view tips that it's trying to show me. So if we look at this process of co-translational protein folding, there is evidence that as this protein is emerging on the ribosome, it will recruit these other proteins called chaperones that will actually potentially shelter the protein. And these play a huge role that is not entirely understood. Sort of what are they doing? Are they just sort of passively holding the protein there, preventing it from crashing with other proteins? Or is it actively promoting folding? Both are viable hypotheses in the field. Probably it depends on which chaperones. And it's an excellent question. And it's sort of an added layer of complexity, but very interesting. Your second question, also an interesting one that I've thought a bit about during the end of my PhD about, different proteins have to go to different places in our cell, right? Some of them, for example, have to go to a organelle called the endoplasmic reticulum, and those are proteins that actually need to be secreted. For example, viral proteins, the proteins that make up the coronavirus, all go through the endoplasmic reticulum. I did some work in my PhD showing that, it turns out that going into the endoplasmic reticulum is also a co-translational process. So as the protein is building up, it gets secreted. And I did some work showing that for a particular protein in the coronavirus, the fact that this kind of entry into this organelle is sequential, happens one amino acid at a time, helps one of the coronavirus proteins fold more efficiently than not. So if you're interested, I can show you. It's a paper in Biophysical Journal 2023. You could look up Bitran and Park et al. But I can point it out. So it's an excellent question that it's sort of an extension of what I was talking about. Yeah.
Great talk. I really, really loved it. So I have two questions. The first is I was really interested in this idea that you can change the properties, some of more of the macroscopic properties of the protein by folding. So you mentioned hydrophilic versus hydrophobic, maybe being greasy or not. And I was wondering if that ever, that change of property ever ends up being an impact for the disease. If that's what allows some of these proteins, the misfolded proteins to be able to accumulate, for instance, would be that macroscopic.
So you're sort of, to make sure I'm understanding your question, it's sort of if you change the overall, the average hydrophobicity or the average amount of charge, does it affect folding? Yeah, that's a great question. And definitely, I believe the answer is yes. So we can make different types of mutations. And in particular, one of the things that is most likely to affect folding is if you look at the three-dimensional structure of a protein, I'll just go back to this slide, right? You can see that many of these proteins have an inner portion and an outer portion. And it turns out that in that inner portion, you often want the amino acids to be hydrophobic because that provides a driving force, an energetic driving force for the protein to sort of fold. And mutations, particularly in that pore, that change the sequence from being hydrophobic to hydrophilic are very commonly types of mutation that actually prevent folding from happening, correct? So things like that, that's sort of one of the types of rules. There are others as well, that you can think about kind of intuitively about what would dictate sort of whether proteins fold correctly or whether a certain mutation, including a possibly disease-linked mutation, kind of would, misfold and cause problems for the cell. Yeah.
So I've heard that some proteins can get tied into knots.
Yes,
How
that is true.
does that happen with the structure you laid out?
That's a great question. How does the protein get tied in? There are, I use these knitting analogies of knots, but it turns out there are proteins where, as Lizzie pointed out, they do form knots, literal knots. How they form is not well understood. Definitely, most likely chaperones, which they were mentioned, will help with this process. It's not something I have a ton of knowledge about, but it's something I'm very intrigued about, for sure. But it's definitely from a topology perspective, from the perspective of forming a structure, it's going to be very difficult. So some help is likely going to be required.
Are there ever kind of, I don't know if this is the technical term, but like weakly folded proteins or proteins that can't hold their structure after they're created or might change, basically like adaptively change or
See,
something?
really cool question. Yeah, so Lauren was asking if there are actually proteins that are dynamic and can actually swap between different structures. And the answer is yes, and it happens in multiple levels, right? So the most extreme case is what we call intrinsically disordered. So it turns out that a substantial subset of proteins, especially in higher organisms like humans, are actually break everything that I just told you. Like everything that I just told you is wrong for those proteins. They're just a big piece of like floppy spaghetti that's constantly changing its shape and doesn't have any structure. And it's thought to be very important. These intrinsically disordered proteins might play a role, for instance, in kind of bringing together different interaction partners and forming kind of hubs for signaling proteins, in sort of, all sorts of functions like that. That's the most extreme case. There is also a bit of a more nuanced case, which I think was actually what you were getting at, which is proteins that don't occupy like all configurations, but occupy a discrete number of configurations. These are really fascinating proteins called fold switching proteins. We don't yet know how many of them exist, because it's kind of hard to detect when proteins kind of swap from one fold and rearrange into another fold. One of my colleagues whose work I'm a huge fan of, Lauren Porter at the National Institute of Health, is trying to develop tools to actually probe how often this happens. And I'll give you an example that one of the proteins that it mediates circadian rhythms in certain bacteria, likely works in a similar way in higher organisms, is known to be a fold switching protein. And the fact is that it actually takes on average about 24 hours for the molecule to actually change from one configuration to the other. And this serves as a timer that helps the organism actually keep track of time. So yes, this can be a functional thing. Is there any Zoom questions? I don't think so. Right?
Yeah, no questions on Zoom tonight, not for your talk either.
Okay.
Let's give Amir another round of applause.
Thank you so much. These have been two excellent talks.
Thank you for hosting.
Yeah, and thank you for this excellent little process as
Oh,
well.
anyone want for? I'm sure you all got it 100% correct.
Yeah.
I'm just gonna quickly,
Oh, yeah.
Thursday
of the month, starting at 5:30.
Oh, no, I forgot the answer.
Oh, it's fine.
It's okay.
I thought it was a catalyst.
So thank you again to our speakers for the talk tonight, and I'm going to just share.
That makes sense.
Okay, sorry, I'm just gonna share some information about our October seminar, which is on the third Tuesday of October, October 15th, same time, same place, and same Zoom link that we have. And so we'll have a talk in astronomy and vision science. And if you're interested in joining our mailing list, that's the QR code and also the shortened URL. Yeah, thank you all for being here, and post this seminar. We will upload the video to our YouTube channel, and we'll send out some more information, including Q&A with our speakers and some resources. So yeah, thank you all for being here.
Thank you.